Okay.
Awesome. Okay. Yes, thank you, Ali, for that introduction and for Victoria for that reminder. I'm going to share my screen. Can you see my screen? Good. Okay. I'm just going to rearrange some windows. I can see all of you. Okay. So yeah, okay. So thank you everyone for attending. Really looking forward to providing a pretty high level overview of the work that my lab does here. So we started relatively recently, about a year and a half ago. And so I've been working on writing grants and getting rejected from grants and trying to hire people and fill my team up. So we're over in the sixth floor of the new Anschutz Health Sciences building in the Center for Health AI space. And yeah, I think like Ali had mentioned, I want to emphasize that if there's any questions, feel free to stop me at any point if things aren't clear or you're particularly interested in a specific slide and you want to learn more detail. I'm happy to do that. I don't need to go through all of my slides today. I want this really to be kept at the most informal that is helpful for getting the message across of what our lab does and what we hope to bring to campus. So I guess without further ado, I'll just get started. I'll jump in. This is the data that we typically work with. So these are pictures of cells at 20x magnification. These are the same cells in 5 different imaging channels plus a merged representation. And so this assay is specifically called cell painting.
Oh, she's good.
So the cell painting assay is going to stain for a bunch of different organelles. It looks like we have some feedback.
Fairly well right now, so.
Yeah, okay, sounds good. So yeah, these stains are unbiased stains. They're going to mark general cell structures, and I'll describe this assay in more detail. I just wanted to give a broad overview of the type of data that my lab works with. But before we actually get to describing the data, I think it's important to set the stage first about how the data are generated and the experiments that we need to perform in order to get the best possible data set that we can analyze. So if you were to come to my lab and you had a particular interest in performing a drug screen, these are the types of questions that we would talk through. So I'd want to know what your overall goal is. I would want to know what your target is. But here, this is a trick question that I tend to ask every once in a while. But I'll describe why this is a trick question later on. And then, you know, if things seem like they're aligning, then I think it's worth sitting down and designing a drug screen together. And the first thing that we would do when we would design this screen is make a bunch of different decisions and essentially narrow the scope of what we're looking for. Drug screens are really broad, but they're only powerful if you get really narrow in your focus and broad in other directions. But essentially, we would want to know what cancer you're, like, what's the context that you're interested in? What assay do you want to perform? There's a lot of different considerations here. Most often we use cell painting. It's a microscopy-based assay, which I'll describe in more detail a little bit later. But there's other choices. And we also need to choose what perturbations we're going to use. And this is an important consideration. There's a bunch of different types of libraries that we might be able to screen. So just a couple examples. My lab is interested in pediatric cancer primarily, but you know, like Ali had mentioned, our techniques are broadly applicable to a bunch of different cancer contexts. But we might choose to focus on neuroblastoma. We might use the cell painting assay, and we might decide to screen a library called the drug repurposing hub, which is a bunch of clinically relevant, therapeutically relevant molecules that are in the clinical pipeline or have already been FDA approved. The next thing after deciding the context and the assay and the library is to carefully design the experiment. So we need to keep in mind different things like cell culture conditions, how we're going to plate cells and treat them, and what type of microscopy are we going to perform. And so to make this a little bit more concrete, Some examples of this could be, maybe we just want standard media that these cell lines are happy in. For the plating of cells, there's a couple considerations that we need to make. So I actually have a prop here that I would have passed around if I was in person, but this is a 384 well plate. This is the size, so it's smaller than my hand. And each of these individual wells is 384 of them. And what happens is in the experiment, usually it's a robot that actually puts the cells within each well and then goes along and pipettes, or it's actually some really interesting technology that you can use sound to vibrate in different treatments at different concentrations to treat each of the cells that are within each of the wells. And then microscopy is performed on this. And we do this in really, really high throughput. But there's a couple of considerations to make when you're designing your plate map, and then also deciding the microscopy. So like I had mentioned, the microscopes these days are mostly automated, so you don't have to stand there, with the pipette and go well by well. Sometimes you have to, but more or less they're automated. This is an example of one. So these are the size of rooms that a scientist can actually walk in, and there's like robots that are kind of moving all over the place. This is called the G3 Explorer. It turns out that Anschutz has one of these, and it's had one for a couple years now. This is built by Perkin Elmer. So the School of Pharmacy just received funding to put this up, and we know the folks over there that are regularly performing these high throughput experiments that maybe some of you on this call have actually used. So to summarize, there's a lot of different experimental design decisions that go into getting the data to use. You need to decide the biological material, decide the assay, decide the drug library. After that, we need to plan for data collection, which involves deciding what are the culturing conditions for the cells. What are the cells themselves too? I guess that goes under the biological material side. We need to decide different cell plating and treatment strategies of plate design, and how to perform the microscopy. And so after that, that's the experimental plan. Then we can decide the analysis plan. And that's where my lab, really my lab should be involved in the entire experimental design process, but my lab is a dry lab only. So we really focus on performing the analysis, and we'll come up with an analysis plan together as well. So that's really the setup. The next part of the presentation is to talk through the data in more detail and how we actually analyze it. But maybe I'll stop there to see if anyone has any questions so far. I think we're about 20 minutes in, so maybe folks are finishing up their last bites of food, but feel free to jump in or put anything into the chat if there's any questions. So it sounds like there's not, which is fine. Feel free to interrupt as usual, but I'll move on now. Okay, so there's a lot of different types of assays that we can collect and data that we can collect, but I think it's worth focusing on just one, which is what our lab primarily has expertise analyzing. And so this assay is called cell painting. And so you can kind of think of cell painting as a color by number, but for cells. So these are unbiased dyes that aren't going to mark any particular gene or protein or cell surface receptor or anything that's specific to the cell. They're going to mark general cell structures. And so the specific dyes in the canonical cell painting assay I have shown over here. So these are dyes that are going to mark the nuclei of cells. They're going to mark the ER, or the endoplasmic reticulum, the mitochondria, actin, plasma membrane and Golgi multiplexed in the same channel, and nucleolus and cytoplasmic RNA. And so these are kind of well-established dyes that have been used for about 10 years. And they're actually extremely cheap to acquire, and that's primarily the basis for their selection. We've worked with a lot of different collaborators to modify this assay in various ways, such as including maybe a cell type specific marker or some other marker that might be more important to the system that we're studying, like maybe BODIPI if we're looking at adipocytes. So applying the stains to the cells, these are the images that we end up extracting. So these are cells. I don't know what type of cells these are, but you can see the different structures that they're marking in unbiased ways. And so these images don't come merged together. You kind of just merge them to make pretty pictures. They really come in 5 separate channels. And so like from here, we see the nucleus staining, the ER staining, the nucleoli and cytoplasmic RNA, AGP, and mitochondria. And these come in 5 different channels. And essentially, we analyze each of the channels individually. And we also merge them for other purposes. But more or less, you can think of them as individual channels that we analyze. So this is the broad overview of the steps, the analysis steps. So really, this could have been my first slide if I didn't talk about the whole experimental design procedure. And I'll walk through each of these steps in relatively good amount of detail for each of them. The first one is to perform quality control and image correction, and I'll talk about what that means. We also perform single cell segmentation. We extract morphology features from all the single cells. And the wheelhouse of my lab is the software. So we develop the bioinformatics software. Not all of these. So Cell Profiler was developed in Ann Carpenter's lab. That's the lab I did my postdoc in. And Deep Profiler is a deep learning version of Cell Profiler. My lab develops PyCytoMiner. And we also develop 2 other software tools called Cytotable and Cytosnake, which I'll talk about in a little bit more detail. And all of this bioinformatics occurs generally in the cloud for drug screens because the data are typically really large. I'll also talk about one downstream application if I have time. I'm going pretty slow right now, so I might, oh, actually, no, because I started 15 minutes early, this is the time up there is really only 10 minutes. So I think I'm pretty good. Maybe someone can keep track of time for me and let me know if like I'm doing okay.
Yeah, you're good. You are
Okay.
15 minutes in right now. So
okay, that's what I thought. Okay, I can do math. So that's great. Yeah, and so yeah, if I have time, I will talk about one downstream application that we do with the drug screen results that we extract, and then I'll talk about some future directions for my group. Okay, so the first step is quality control and image correction. I have a pre-image on the left and a corrected image on the right. I'm going to ask for audience participation here. Does anyone see the difference between the image on the left and the image on the right? And can anyone tell me what the difference is?
Well, I see a color difference.
Yep. There's
Yeah,
definitely
I
a
feel
difference.
like there's more green, sort of glowy green on the left.
And
Yeah.
purple.
Yeah. So what correction would I have applied? That's a harder question.
We have contrast is different in the chat box
Yeah.
here. Yes, George.
Okay, yeah, that's great. So this procedure, what we're doing is adjusting for this phenomenon called vignetting. So this is in a microscope. You take a picture, and it turns out that the illumination along the edge is darker than the center. So the light shines in the middle, and it diffuses towards the edge, and you don't get as bright edges. So that's all we're doing here. We're just adjusting for the uneven illumination. It does more than just adjusting for the vignetting. It does other things with, I guess I have some slides to talk through that. But essentially what we want is when we're analyzing these images, we don't want any cell that's in the corner of the field of view to be any different artificially from a cell that's in the middle. So we need to adjust these sorts of technical artifacts, or else there's not really any hope for us. So this is more broadly, the procedure is called illumination correction. So this is a computational procedure where we take our original images and we adjust for uneven illumination in the background. And so you might look at these 3 pictures. These are Schwann cells now, 3 different stains. And you might look at these initial images and be like, yeah, they look great. But if you go and actually adjust the brightness to them, you can see that they don't actually look all that good. And what looks good to our eye is almost guaranteed to be very different to what a computer sees. So we use a computer to analyze all these images. We don't do this by eye. We generate billions of single cells, so we can't possibly do it by eye. So we need to have a computer that is going to be able to adjust these types of things. So if you brighten them, you can see that there's like really these shadow effects by these cells. You see even in the RFP channel, it looks like there's maybe like white noise almost from like an old television set that's not tuned to a particular channel in the background here. And we want to adjust for them. And so we perform a procedure called illumination correction, or IC, to adjust for these types of background artifacts that we can't necessarily see by eye. Maybe there's a
We have a
question in the
quick,
chat.
yes, we
Yeah.
have a quick question. So basically George is asking, so basically magnified the view of those hidden cells, and that was on the slide with the colors and back, yes, that one. Asking if magnified the view of those hidden cells.
Yeah. So magnify is a tricky word because magnification is a specific thing with microscopy, but essentially, like, you do want to make sure that you extract the features from the cells that are along the border in the same way that you would extract them from the middle, because there's no difference between them. There shouldn't be a difference between them. The only difference that we would pick up on is a technical thing. And we want to avoid picking up on technical things. Reducing the signal from the technical and amplifying the signal from the biological is always the challenge with computational biology. So now by eye, which is still dangerous to do, but you can see clearly that illumination correction has had an effect where the background is now uniform across the different channels. And I think particularly interesting is the RFP. So not only did we adjust for this strange granularity in the background, but we've also really popped the structures. An RFP in this case is tagging actin. The next step is a really important one too. It's called segmentation. And so this is it's actually a pretty buzzword in like tech right now, because both Facebook and Microsoft just released like their large. So ChatGPT, I think most people have been exposed to and have heard news stories, if not used it themselves in their jobs. One of the next areas of expansion for these types of models is in computer vision. And there's a lot of research already on this. It's not like, that's not in like particularly mind-blowing statement, but there's a lot of interest in segmentation, particularly in like the avenue of self-driving cars, for instance. So what we want to do is we want to be able to see where things are. We want to be able to identify what's in the foreground, what's in the background, and in this case, figure out what's a car, what's a person, what's a bus, what's a traffic light, what's a tree, and so on. So we apply these similar algorithms in our case, but instead of segmenting people or cars, what we're doing is we're identifying the boundaries of every single cell that's in our image. And so this is an example. It's kind of granulated because I increased the zoom on this, but essentially one thing that we do is we segment the DAPI channel, or the nucleus. And so all of these are nuclei. We're able to segment them, identify the boundaries relatively easy. This is a relatively straightforward problem because it's basically dots in a black background. And so it's not like a particularly hard algorithm. It does get trickier, especially in like these cells that are spindly and might be overlapping. So these are Schwann cells, which are neuronal based cells that are going to have these types of structures that might be difficult to segment. You might argue that there's actually no correct answer here. So like, for instance, we can apply a segmentation algorithm, and the segmentation algorithm might tell us that this right here is the boundary between this cell and this cell. But it could have just as easily made the boundary just like a millimeter or a micrometer higher. Like sometimes it's obvious, sometimes it's not. And we have procedures to basically evaluate how well segmentation is performing. So it gets slightly more complicated than just looking at the individual nuclei. Here's another example of different tools that we've benchmarked and are continuing to benchmark their ability to segment these more complex structures. And this is also only working in cell line. There are folks that develop algorithms for segmenting cells in tissue, which is much more complicated, and the cells are all sorts of different shapes and sizes, and defining boundaries is really difficult in tissue. Slightly easier in cell line, but we still have similar problems. So it's a matter of selecting which method and tuning some parameters about how to optimize some of these single cell segmentations. Segmentation is also slightly more complex. So I'll go one level deeper in terms of complexity. So we segment two different structures. We segment the nuclei, like I mentioned, and the whole cells, like I'd also mentioned. But we also do a subtraction. So most cells, not all, but most cells have one nucleus, and essentially we perform a subtraction. So that subtraction will give us the cytoplasm. So it's basically the cell minus the nucleus. And we use Cell Profiler most of the time to perform this segmentation. And this is important because in the next step, when we perform feature extraction, we perform feature extraction from each of these types of segmentation, which I go to in this slide. So we generate masks. So these are just, is this in the foreground or is this in the background? We apply these masks. So 3 different masks across the 5 imaging channels. So 3 different masks, 5 imaging channels. That's 15 different per single cell entities or objects that we're measuring. And so with those 15 measurements, we end up generating between 1,500 and 3,000 features per single cell with the canonical pipeline. And so these features, we also typically use Cell Profiler for this. That's really been our workhorse so far and will continue to be for the foreseeable future. We're also actively working on improving some of the representations using deep learning, but Cell Profiler is a tried and true method. So within these 15 different categories, we're measuring each of these feature extraction categories. So these are things like geometric measurements. What is the area of the cell? What is the perimeter? What's the maximum radius? Things like that. Oh, maybe there's a question.
Yep. Grace has a question.
Great.
Yeah, thanks. So back on that slide about the subtraction, I'm just curious if you perform that subtraction on the nuclei so that you get a clearer look then at the organelles. Is that essentially why?
Yeah, yes, exactly. So if I go maybe, yeah, here. So that's exactly right. The nucleus is more or less gonna, there are going to be some organelles that are in the Z plane. So we do measure some mitochondria that look like they're in the nucleus, but because the cells aren't 2D, they're 3D, some of the mitochondria could be on top. But performing that subtraction really allows us to focus on the ER and the mito and the Golgi that we expect to be outside the nucleus.
Okay,
that's a great question.
Thank you. Yeah.
So yeah, like Grace had clarified, the purpose is to extract these features from the different imaging channels, so that these features will be able to inform and tell us something about the ER or about the mitochondria. And what they're going to tell us about them are things like geometric measurements, texture-based measurements. So in the case of actin, for instance, are all the actin fibers going in the same direction, or are they kind of more or less distributed and there's no real seeming, maybe there's some disorder with the actin fibers. We also measure granularity. So how many spots are present? What's the distribution of the intensity patterns within these spots? We also look at intensity measurements broadly. So how much intensity is present for a particular channel in that cell. So if it has high mito intensity, there's a lot of mitochondria activity that's going on. And we can also look at different sections of intensity. So is there a lot of intensity around the plasma membrane, or is it really localized to kind of close to the nucleus? We also measure other distribution-based measurements. So are things kind of pushed all the way to one side? Does it seem like there's like a polarity to the cell where a lot of the intensity patterns is just kind of pushed towards one side of the cell, or is it more broadly distributed? And there's lots of nuance to these measurements as well. These are kind of the broad categories. And like I said, we extract about 3,000 of them per single cell. Just a slide here to note that the cell profiler-based features are really the canonical computer vision-based features that have been developed over decades of computer vision research. Cell Profiler is a tool to implement those computer vision mathematical functions. We're also actively developing, in collaboration with a lot of other folks, deep learning-based feature extractors. So this is built initially by Juan Caicedo, who is a postdoc with me at the same time in Anne's group. And there's also emerging strategies that we're hoping to incorporate into Deep Profiler based on other deep learning models. So in this case, this is a recent paper that just came out where it's from the Snyder lab at ETH Zurich, where they trained a vision transformer. So a transformer model is the T in GPT. So applying a transformer model to these types of data, it's still relatively unclear how better these representations are compared to the classical computer vision features. The hope is that they are going to tell us more specific things about the underlying biology, but I guess it's still out whether or not they are outperforming some of the classical vision features. Okay, so now we've been able to extract about 3,000 features using the classical approach, and we'll just stick to the classical approach for now. 3,000 features per single cell in these drug screens. What we need to do now is to normalize these vectors. So you can imagine now we have a 3,000 dimensional vector, and what we need to do is make sure that that vector is behaving nicely, and that we can apply statistics and machine learning to that vector. So that's where PyCytoMiner comes in. I'm not going to go through all of these, this complex, busy slide, but PyCytoMiner is the tool that we develop and use to essentially convert that readout to something that we can play nicely with. So we're also now butting up against what's currently state of the art as well. So you can imagine in drug screens where we're screening thousands to hundreds of thousands, maybe even 1 million compounds in a single experiment. That is a ton of data. And right now, the current state of the art is to take every single cell in your image and aggregate them to form one single data point per image. So that is essentially ignoring all of the single cell information. You still apply the segmentation and you still extract features per single cell, but it's really washing away a lot of information. And the Waylab here thinks that you're washing away so much information, why not use the single cell data? But the reason why people haven't used this, and this is common in industry and in academia, is that there's existing software just can't handle it. And there's not methods, like there don't exist methods to analyze single cell data. And so we're developing 2 tools actively right now called Cytotable and Cytosnake, which are going to enable single cell analysis and also enable reproducible analysis. So a lot of times these drug screens, because it's just so hard to deal with the data, have a kind of a standard pipeline that a scientist will apply and extract insights from. But then if you try to go back to that 3 years later, or even 3 months later, it's often really, really challenging to figure out what actually was done to the data to get it to the point where we're at. So we're trying to solve those types of problems with software in the lab. I should also mention that all of these software is open source, and anyone can contribute to it and see the code. We also rely on cloud computing. It's more or less required to analyze drug screen data. Luckily, Alpine exists. So Alpine is the large cluster computing resource that we have access to on campus. It was also recently released. It's a really powerful machine that subsidizes the cost for CU researchers. So you can think of this very similar as you would with some of the commercial cloud providers like Microsoft Azure, AWS, and Google Cloud. So there's lots of things that you can do now that you have. So yeah, let me take one step back. So I've talked about like where the lab is butting up against state of the art. Now just considering the canonical pipeline where you take all of the single cells and you aggregate them together. So you form one data point per image. And so you're just focused on that and also cell profiler features. Now we're back to that realm. If I have time, I'm gonna talk through one application, which is a drug screen that we performed during my postdoc. Maybe there's something in the chat too. Oh no, okay. Still two. I just didn't reset it. So this is where I talked about the trick question. So before any of this happens, we need to really think through what the target is. Is your target a gene? So if you're looking at a particular cancer type and you think that this gene is really important to target with a molecule, because if you can target this gene, you can reduce its expression. And it's been shown that if you reduce this gene's expression, you might have a good chance of killing that cancer cell. It's this concept of synthetic lethality. Or is it a protein complex? So not necessarily you're targeting a gene, but this complex forming. What is the best target of this complex to make sure that this complex doesn't have function? Or is it some other biomarker that you're interested in? So this is now butting up against what's currently thought and state of the art. But really, we like to think of compounds not necessarily as targeting one very specific thing. It's going to do a lot more in a cell than maybe we anticipate, and certainly going to do more in a cell in vivo than it is in a dish. But we want to essentially let the data do the talking. And so thinking about a compound more as like a set of keys that might unlock different doors in different contexts is probably a better analogy than thinking of precision medicine, of looking at an arrow that's going to target right in the middle of that bullseye. So what we encourage folks to do is to define some drug discovery biomarker that's phenotypic, not genetic. So the example that we give is, and this is something that we're actively pursuing now, in NF1, NF1 is a haploinsufficient disease. And what that means is that one of the copies that you have inherited from your parents doesn't make fully functional neurofibromin, or doesn't make fully functional protein. And so in NF1, if you don't have anything, it's if you're developing embryo, it's a lethal phenotype. But if you've inherited a sporadic or a germline mutation in NF1 from mom or dad, you're not producing enough neurofibromin to have normal neurofibromin function. And this is the category of disease called haploinsufficiency. And so what we're trying to do actively is to develop experiments to model what haploinsufficiency looks like phenotypically, so that we can perform a drug screen to specifically reverse that phenotype. And I think that's for these particularly challenging diseases that, cancer, for instance, that don't currently have cures. I think really focusing on that phenotypic biomarker is going to do a load of good for making sure that your drug screen actually gets some hits that we want to validate later on. That might have just been me on my pedestal for a little bit. But yeah, so that's basically the gist of it. And this is the end of portion 2 of the presentation, where we perform image quality control, we correct the images, for instance, in illumination correction. We segment every single cell. We extract cell morphology features, most often using Cell Profiler, but there are also emerging representation learning algorithms that might be able to do a better job of describing cell morphology. We apply a bioinformatics pipeline and other various pieces of software to process the images and process the cell morphology features that we've extracted in preparation for different downstream analyses. And so putting the entire experimental design into context on this one slide, we start with our cells of interest. These are cancer cells, or these are healthy cells, some sort of disease. We perturb them with a drug library or genetic perturbations, any sort of way of changing the cellular phenotype. We plate these cells in high throughput in 384 well plates. We apply an assay, most often cell painting or some modified version of cell painting, which are going to mark various different organelles. We apply image analysis tools and software to segment cells, extract features, and normalize them and get them ready for downstream analyses in which we can test various hypotheses. And so that's it for part 2. Like I did with part 1, I'll pause to see if there's any questions, and also ask if I have enough time to talk through my application, or if we should pause here.
You're doing great on time. I actually have a question for you about the segmentation.
Okay.
When you showed those images, you had one where it was like your own segmentation and versus the different technologies that you could do.
Yeah.
Are these things that are being developed, like how important is that, the accuracy of that?
Yeah, that's a really, really good question. It's unclear how important it is. There haven't been extensive benchmarking performed to really figure out the impact of segmentation. It seems obvious that it should be really important, but at the same time, until we have data to describe how important it is, we don't know. A lot of times we just kind of do what tends to work, and we check a bunch of images by eye and see if the segmentations look good. And then we kind of stop there, at least for now.
Okay, cool. And I think Eric had a question as well.
Yeah, you kind of answered it there. I just wanted, when we were talking about segmentation, you mentioned evaluating how good the segmentation is basically. And I was just wondering, what is the method for that? Is it completely done by eye, or because it seems to me that, if you wanted to develop like an algorithm to check that, the algorithm would need to be, quote, more accurate than
Right.
whatever method you're using.
No, exactly. It's a really important insight there, that yeah, you really have to rely on ground truth when you're doing evaluations. And for segmentation, almost never do you have ground truth unless you're running like a simulation where you know exactly what the boundaries are because you've preset them. But yeah, typically what's done is you'll segment something by hand. So there are tools out there that you can like individually outline a cell and then measure basically the overlap between an algorithm like CellPose or Cell Profiler and the hand-drawn segmentation, where you kind of consider the segmentation that's hand-drawn to be ground truth. But like I said, it's really tricky.
Thanks.
Okay, I'll talk through an application. Okay, so like I had mentioned, the first step in a drug screen is to design the experiment, and so we've designed this experiment. It's actually done before I started in the lab. I inherited this project, but this is my postdoc lab. But essentially, we chose to analyze A549 cells. So these are lung cancer cells. And we hand selected 1,327 different drugs from the drug repurposing hub and measured them in 6 different doses. So this ended up being about 250 of these 384 well plates. And we collected assay data. But what was important about this particular experiment was that we collected multiple different kinds of assay data. So 2 different assays. L1000, which is a gene expression assay, and cell painting, which is a cell morphology assay. And so essentially we applied the respective bioinformatics pipelines to process the data. And now we have our L1000 data and our cell painting data that we're able to compare apples to apples. And what's important about the design here is that we use the exact same plate layouts. They're different cells, but the exact same plate layouts to measure the gene expression and cell morphology. And we did a number of things in this experiment. But one thing that's really important to do is to determine how reproducible measuring that compound perturbation is. So ideally, if you treat cells with a drug and you do that in 4 different wells or 5 different wells or 20 different wells, you want the measurements that you're extracting from that well to be consistent. That's a good it's a measure of a good assay if it's measuring the same perturbation consistently. And so this might be meaningless, but essentially you can think of the measurement as this high dimensional space that exists in this landscape, this epigenetic landscape that was, this idea is basically the founding of systems biology by Conrad Waddington in 1957. But you can think of this landscape as biology is constrained to move in only certain regions. And if you measure biology, you're likely going to measure it in one of these basins of low energy that just kind of collapse into this high dimensional space that's more or less a stable space. And so the way to represent this as in terms of reproducibility is that every time you measure that drug, you kind of want to be around that high dimensional space, around that region of lower energy. So if you have a measurement like this in this drug, you measure it 5 times, that's probably a pretty consistent assay. But if you measure it and it's all over the place along this high dimensional landscape, it's probably not a good thing. And either the drug's not doing anything, and so therefore it's kind of a random response, or there's something going on with the ability to extract features. And so we did this based on that experimental design with 1,327 different compounds. And so this is going to be maybe my only data slide, or I have 2 data slides. But what the y-axis represents is how many compounds were, you can think of it as being reproducible, but we had to kind of artificially define that. So if you randomly sample non-replicates and form a distribution, how many of the compounds are greater than 95% of that curve? And so that's one way of saying that we're more reproducible than by chance. So it's kind of a low bar, but still what we see is cell painting, which is in yellow in the lowest dose here, measures 582 different compound perturbations that L1000 doesn't capture. So L1000 captures 68 unique, and there's an overlap of 133 for a total of 62% of that 1,327. And so unsurprisingly, as you increase the dose, you increase the reproducibility all the way up to 10 micromolar, where you get 85% of the compounds you measure reproducibly. But still, consistently, cell painting tends to outperform L1000 in terms of reproducibility. One of the other things that we did was to assess this idea of mechanism of action. So a lot of drugs have been annotated to a specific mechanism. They target this one very specific cell surface receptor, and that's what they do. They were designed to do that. So they've been pre-annotated. And because we're using drugs from the drug repurposing hub, these are relatively well-characterized drugs, and many of them have at least one mechanism of action. Sometimes drugs have multiple MOAs. But just like in the previous experiment, if you have drugs that all have the same mechanism of action, this pentagon here, we want an assay to reliably measure them similarly. Whereas if they're kind of all over the place in this high dimensional landscape, it might not be an effective assay. And so you can do this exact same visualization where at the low dose, I think there was something like 50, somewhere between 50 and 80 different MOAs. I don't remember the exact number, but we captured 20% of them with both assays, 16 uniquely with cell painting in the lowest dose. So unsurprisingly, again, as you increase the dose, you get more coverage in your MOAs that you're able to reproducibly measure. But still consistently, cell painting tends to outperform, although there is more overlap between the assays. This is another way of looking at that might be more specific and interesting to look at, because now we're looking at very specific MOAs. And so what immediately jumps out is, sorry, I should describe the axes. So the axis here is some level of performance for the L1000 data, so gene expression. The higher you are on this, the more gene expression captures that specific mechanism. And the x-axis is the same metric, but using cell painting. So anything along the diagonal are going to be things that you measure very similarly between gene expression and cell morphology. And anything off the diagonal is things that might be measured better with L1000 or better with cell painting. So things like purine antagonists, you can predict them, or they have really, really strong reproducibility in cell painting, but there's no signal in gene expression. Or things like HSP inhibitors have really, really strong signal in gene expression, but they have no signal in cell painting, which was really interesting to us. Also interesting is that most mechanisms we can't predict. So most everything kind of clusters towards things that are just seeming like they might be random. I think I'm going to skip this part. We did this in a more formal deep learning sense, where this was more or less an unsupervised approach, where we just looked at the high dimensional space and compared same compounds to one another, and also compounds from different mechanism classes. Here, we actually did perform supervised prediction, where we took very specific models from this previous Kaggle competition to actually perform predictions. And maybe I just will go through this really quick, but you can evaluate predictions in this sense at per MOA level. So this is a particular figure called a precision recall curve, where you want your curves to be pushed to the top right. And so the further you are to the top right, the better the performance. And so this is an example of a particularly well-performing model using cell painting readouts. We can predict EGFR inhibitors relatively well across the different models that we trained. You can also look at L1000, and basically for all of the, for every single MOA, we can perform this type of prediction. Like MEK inhibitors are really have really strong signal. We can predict them really well. We can predict mTOR inhibitors, and so on. This is a greater, so this is only 3 examples on the left. These are all of the MOAs on the right. It's very similar to the plot that I went through, where we see off diagonals, but also a lot that are predicting really well, and the majority of them that have no signal at all. So essentially the take-home message of this paper was that you can measure genomics, so the pulp in the insides of the cells, or you can measure cell biology, the fruit stands, the shape and size and texture of the cells, and you get different information. And so the moral of the story is, if you're doing a drug screen and you're looking for compounds that might be effective in a particular disease like cancer, why not measure both? I mean, the answer is often economics, but cell biology, I think, is an emerging strategy that is worth exploring in more detail. And so we did all of these analyses on these rows, and basically there's a trade-off between cell painting and L1000, so cell morphology and gene expression. And there's lots of different considerations to make when designing when deciding which assay to pursue. This was a cell systems paper that was published, I guess it was about a year ago. And these are all the authors on the paper. It was a big team effort led by Anne, Aravind, and Shantanu, and also my co-first authors, Ted and Adeni, were on this paper as well. All of this is publicly available. So the software to reproduce the full analysis is there. All of the data are publicly available on a resource called Image Data Resource, or IDR. So this is, I think it's like 43 terabytes of images. And also the process data are available as well. So just one quick slide on some future applications that we're developing right now in the WayLab. So we're writing a bunch of grants to do these things, and are getting started to do them anyway. Essentially, we're defining a pediatric cancer cell morphology atlas, which we think will help with finding cures for pediatric solid tumors. We're also developing the cell painting assay and analysis pipeline to perform high throughput drug screens in patient-derived organoids. We are also, like I'd mentioned with the NF1 example, we're hoping to soon design and implement a drug screen to reverse NF1 haploinsufficiency in Schwann cells. And so that's all I have. Thank you for your attention. The lab is currently growing right now, so we have a lot of really awesome interactions with the team. So Eric, Jenna, Roshan, Dave, and Mike are the full-time members right now. Mike is a rotation student. And there's also a bunch of alumni in the group. And yeah, thank you for your attention. I'll take any questions.
Thank you so much, Dr. Wei. And if anybody has questions afterwards that you think of, feel free to let us know. We can pass those on. Like I said, we'll be sending out those certificates to everybody. And Dr. Wei, I'll send you some data so you can see who was able to attend.
Okay, awesome. Thanks again.
Thank you all.
Thank you.
Thank you.